Good afternoon. My name is Hui Guanlin. Currently is a professor in Duke University School of Medicine. Today I have privilege to introduce keynote speaker, Dr. Yosuke Togashi. Professor Togashi is actually received an MD degree from Kyoto University in 2006. He then received a PhD in 2015 from Kindai University. Currently, Dr. Togashi serves as a professor in various departments from Okayama University. And Okayama University. And Dr. Togashi actually current research focus on tumor biology and particular interest in the cancer immunology. And he has been focused on various human disease, including the pulmonary biology and also the lung cancer study. So he has made a great contribution in understanding how cancer immunotherapy actually fail during this treatment. And he actually identified several factors cause T-cell exhaustion that actually account for cancer immunotherapy resistance. And today, his talk actually mainly focus on one of very exciting finding where the mitochondria DNA can be transferred from the cancer cell to the tumor infiltration T cell. So today he got to give us a new insight how T infiltration cell could be regulated during the cancer progression and how it actually contribute to the cancer immunotherapy efficacy. Without further ado, please join me to welcome Dr. Togashi for his talk. Thank you.
Thank you, Professor. I'm Yosuke Togashi from Okayama University West Side in Japan. So I apologize for not attending in person this exciting symposium because last night I have my own work in Japan, and tomorrow afternoon I also have my own work in Japan, so it's difficult to go to Taipei. Sorry, today I'm going to present my recent work via Teams web meeting. And my main focus is cancer immunology, tumor microenvironment. Especially, I frequently analyze tumor-infiltrating lymphocytes from clinical sample. So my as previously said, my present work focus on TIL and mitochondria transfer. So I'm going to talk about my recent work, especially mitochondrial transfer. I started cancer immunology research since 2016 after approval PD-1 blockade ICI in Japan. In Professor Nishikawa Laboratory National Cancer Center, regulatory T-cell laboratory. My first motivation is to search to identify biomarker to predict ICI efficacy. Immune checkpoint inhibitor ICI exhibit efficacy by reactivating effector T cell. There are various T cell in the tumor microenvironment, but I think T cells that directly attack tumor cells are the most important because PD-1 blockade activates such tumor-specific T cell. So first, I analyze TIL from patient treated with ICI by a single cell level to search for predictive biomarker and such a tumor-specific T cell. This is simple result of my previous 5 years ago work. Top 2 panel responder TIL analysis data and bottom 2 panel non-responder TIL analysis data. We use cyto maybe with more than 30 parameters, and we focus on this cluster. This cluster was dominant in responder, but not dominant in non-responder. This cluster, very simple cluster, PD-1 positive, CD8 positive T cell. So we thought the PD-1 expression in CD8 positive effector T cell could be a predictive biomarker. And so we next speculated this cluster, this population could be tumor-specific directly attacking effector T cell. We sorted this cluster and we checked tumor specificity and we confirmed such PD-1 positive exhausted T cell in the tumor microenvironment actually directly attack tumor cells. And so I'm interested. I was interested in such exhausted T cell from around here. And we are now analyzing spatial data about exhausted T cell in the tumor microenvironment, especially focusing on tertiary lymphoid structure. We found exhausted T cell cluster within TLS region. Of course, we also found exhausted T cell cluster within non-TLS, mainly tumor cell region. And we found different feature between TLS region exhausted T cell and non-TLS region located exhausted T cell. And now we are now submitting these data. But I previously belonged to Dr. Nishikawa laboratory, and his laboratory originates from Professor Shimon Sakaguchi laboratory. So 5 years ago, my main focus was regulatory T cell in the tumor microenvironment. We also found PD-1 high expressing regulatory T cell in the tumor microenvironment from clinical sample. And such PD-1 positive regulatory T cell could be activated by PD-1 blockade, especially suppressive function. As you can see here, this is controlled no PD-1 blockade, and regulatory T cell suppress effector T cell reduce the cell proliferation to 36%. And if we added PD-1 blockade in this to this condition, effector T cell proliferation reduced to 15%. So PD-1 blockade activated PD-1 positive regulatory T cell mediated suppressive function. So such PD-1 positive regulatory T cell infiltration could be resistant biomarker to PD-1 blockade, as you can see here. So we published these data in PNAS and Nature Immunology, collaborating with Shimon Sakaguchi and Professor Honjo. This paper is my honored paper to collaborate with 2 Nobel Prize winners. Very honorable data. But after 5 years ago, I had my own laboratory. So after my having my own laboratory, I'd like to search for my original research project. And I'm interested. I was interested in T cell exhaustion in the tumor microenvironment. So T cell exhaustion have several features in the tumor microenvironment. Especially, I'm interested in mitochondria metabolic dysfunction in exhausted T cell. So I started mitochondria project in T cell exhaustion in T cell in the tumor microenvironment 5 years ago. What is mitochondria? Very simple mitochondria explanation slide. Ancient bacteria, more than 1 billion years ago, ancient bacteria enter into eukaryotic cell and became organella mitochondria. And this is endosymbiotic theory with eukaryotic cell and mitochondria. So such mitochondria have original DNA, mitochondrial DNA, and mitochondria produce ATP through oxyphos. So based on such endosymbiotic theory, such mitochondria could transfer to other cell from one cell to another cell. This is mitochondria transfer research field. This research field started since 2006, about 20 years ago. Not my first report. This is a first report about mitochondria transfer. PNAS 2006, first report mitochondria transfer, but in experimental artificial model. However, recently there are several published published studies about mitochondrial transfer in vivo, especially focusing on tumor microenvironment. But such data mainly focus on transfer from other surrounding cell to cancer cell, tumor cell in the tumor microenvironment. Such transfer surrounding microenvironmental cell provide mitochondria to cancer cell, leading to tumor progression. There are several recent published studies. In addition, last year, myeloid cell provide mitochondria to T cell, which enhance T cell effector function from Germany group. This is very exciting data. But I'm interested. I was interested in from cancer cell to surrounding tumor microenvironment cell, especially T cell. I wanted to know how about transfer to T cell from cancer cell in the tumor microenvironment. So opposite transfer compared with previous data. So we first analyzed mitochondria DNA using clinical sample because we had paired clinical sample TIL and cancer cell and peripheral blood. So we sequenced mitochondria DNA from such paired sample. Very happy, very lucky. We first sequenced about 10 TIL and cancer cell, and we found about 40% TIL have mitochondria DNA, and many of which shared with cancer cell. This is very exciting, lucky data for me. However, there are several concerns about contamination. This slide shows our TIL establishment procedure, tumor digest and its culture. Mainly our established TIL consists of T cell. However, there are several concerns about contamination. So we checked our established TIL using flow cytometry, FSSS scatter plot, and singlet, live data, and most our established TIL consists of CD45 positive, CD3 positive T cell. We again sorted this population and we confirmed mutation. This is bulk TIL mutation and sorted TIL mutation, and cancer cell TIL mutation and wild type peripheral blood. And we also sequence surgical rejected to formalin fixed sample after rejection because there are another concern about acquired mutation. But we confirmed mitochondria, same mitochondrial DNA mutation in FFP formalin tissue clinical sample. So we concluded our identified mitochondrial DNA mutation were not contamination and not acquired mutation. So we speculate mitochondrial transfer, and we conducted mitochondrial transfer experiment. We stained TIL and cancer cell with different mitochondrial specific marker. We used mitotracker green to stain TIL, and we used specific reporter protein mitoDS red for cancer cell. And co-culture for our 3 or 2 days after 48 hours later, we confirmed red color enter into TIL. Blue color means CD45, and this is this circle figure mean TIL, and we confirmed red color enter into TIL. And so we confirmed mitochondrial transfer from this experiment. What mechanism of mitochondrial transfer? There are several reports about mitochondrial transfer mechanism. We created several conditions, various conditions to investigate the mechanism. Please look at these bar, for example, and this condition we use insertion column, meaning separate TIL and cancer cell. And insertion column reduced mitochondrial transfer. And this, please look at this bar. In this bar, we use cytocalcin B. This reagent, tunnel nanotube inhibitor, as you can see here. And this inhibitor also reduced mitochondrial transfer. In addition, please look at this bar. And this bar we use GW4869. This inhibitor, this reagent is EB inhibitor, especially exosome release inhibitor. So EB inhibitor also reduced mitochondrial transfer. So we concluded TNT direct contact with TNT and indirect EB play important role in mitochondrial transfer from cancer cell to T cell. However, I have one question at this point. There are many mitochondria in a single cell, more than 100 mitochondria in a single cell. However, just 1 or 2 transfer maybe do not affect T cell function. However, in our sequencing data, more than 80% mutation were found in TIL. We call such condition homoplasmy. I have one question about homoplasmic replacement, if homoplasmic replacement occur or not. We checked replacement, green mean till original endogenous mitochondria, and red mean cancer cell-derived mitochondria. Time-lapse imaging clearly show green color gradually reduce and red color gradually increase. Eventually, only red color cancer cell-derived mitochondria remain in TIL. We also confirm mitochondrial replacement, homoplasmic replacement with sequence. before co-culture wild type, and after co-culture 1 week or 2 weeks later, mutation gradually increase and eventually a part of TIL have homoplasmy mutation. We call this phenomenon hijack. This is in vitro data. So we also perform in vivo mouse model experiment. We have mitochondrial DNA mutated tumor cell, lung cancer tumor cell, mouse lung cancer tumor cell. So we introduced mito-DS red in this tumor cell and transplant mouse model, and we analyzed TIL time-dependent manner. This is TIL analysis flow cytometry data, scatter pro, FSSS scatter plot, and singlet live data, and we analyzed this population, CD45 positive, CD3 double positive T cell population. We confirmed DS-led, meaning mitochondrial transfer from cancer cell gradually increase, and 6 weeks later, more than 15% transfer. And again, we sorted this population, negative DS-led negative population and DS-led positive population, and sequence again. And negative population, all negative population had wild type, and positive population, DS-led positive population had mutation. Of course, there are several heteroplasmy, but we confirmed homoplasmic replacement actually occur in vivo. So next, our question, what made homoplasmic replacement? Where have original endogenous mitochondria gone? So we first evaluate original endogenous mitochondria in co-culture condition. Before co-culture, original mitochondria, as you can see here, evaluated by mitotracker green. But after co-culture, original mitochondria, original endogenous mitochondria reduced, as you can see here. This is, we evaluated mitochondria reduction after co-culture. However, such reduction was reversed by NAC antioxidant reagent. As you can see here, NAC reversed the reduction evaluated by mitotracker green. We also confirm mutation, mitochondrial DNA status. Mitochondrial DNA status also reversed by NAC NSC to wild type. So, we speculated mitophagy induced by ROS, antioxidant stress, could be related to such replacement. So we analyzed mitophagy using LC3B staining. After co-culture, bottom panel, LC3B increase, and this distribution similar to green color till original endogenous mitochondria, meaning such till original endogenous mitochondria receive mitophagy by co-culture. However, cancer cell-derived mitochondria red color distribution different from LC3B. So such cancer cell-derived mitochondria resistant to mitophagy. NAC also reduced mitophagy in endogenous till mitochondria, and bapillomycin A mitophagy inhibitor reversed mitochondrial reduction after co-culture. So we concluded difference in sensitivity to mitophagy between TIL and cancer cell contribute to the homoplasmic replacement. To make long story short, I summarize here one summary figure. We focus on USP30 molecule, mitophagy inhibitory molecule, highly expressed in cancer cell. And this molecule attach to mitochondria in cancer cell. And such mitochondria can be resistant to mitophagy in the tumor microenvironment. And such resistant mitochondria transfer to TIL with USP30 because USP30 attached to mitochondria. In the tumor microenvironment, there are a huge amount of ROS. So ROS induce mitophagy to against the original endogenous TIL mitochondria. However, cancer cell drive mitochondria resistant to mitophagy. So eventually, such dysfunctional mitochondria remain and hijack homoplasmic replacement to actually occur. This is our mitochondrial replacement summary figure. However, not all explanation. USP30 cannot all mechanism explain all mechanism, account for all mechanism. We are now searching for additional mechanistic molecule. From here, I'm going to present functional analysis data. Oxphos in mitochondria in T cell play important role in long-living memory T cell formation. And there are several reports about mitochondrial dysfunction in T cell mouse model. And such mouse model induce senescence in such mouse. And such mouse are reportedly sensitive to infection. So we analyze the T cell function using this based on these findings. First, we evaluated metabolism using flux analyzer, CFOS flux analyzer. We created mitochondrial DNA mutated T cell line. Bold cell line mean mitochondrial DNA mutated cell line. And this is control. And as you can see here, for example, mitochondrial respiration clearly reduced by mitochondrial DNA mutation. Dependency on glycolysis increased and ATP production reduced by mitochondrial DNA mutation. We also analyzed the senescent molecule, for example, ROS, P16, and we also evaluated SASP, IL-6, and IL-1 beta. Please look at this red bar. Red mean DS positive transferred TIL, and 02 mean wild type, 04 mean mutated cell line. So please look at this shadowed red bar. For example, ROS clearly increase and P16 also increase, and SASP, both SASP also increase. So mitochondrial DNA mutation transfer induce T cell senescence. And we evaluated the memory formation, central memory fraction and long-lived memory fraction. Central memory reduced and long-lived T cell memory fraction also reduced by mitochondrial DNA mutation transfer. We also checked activation molecule PD1 and CD69. These molecules also decreased after transfer, mutation transfer. We created this mouse. This mouse, conditional knockout mouse, mitochondria, T cell specific mitochondria dysfunction mouse model. And using this mouse, we evaluated PD1 blockade efficacy. Blue color control and red color conditional knockout mouse. PD1 blockade exhibit efficacy in control mouse. In contrast, conditional knockout mouse, PD-1 blockade not effective against this sensitive tumor. We also evaluated memory formation. Initial PD-1 blockade completely reject some of mouse, and we corrected such rejecting mouse, and after 1 month later, we rearrange same tumor, parental tumor in such mouse. Blue color control and red color conditional knockout mouse. All mice reject parental recharge tumor. However, in conditional knockout mouse, only 1/3 rejected and 2/3 grew rapidly. So we concluded not only just anti-tumor immunity, but also such memory formation also impaired by mitochondrial dysfunction in T cell. Finally, we analyzed clinical sample mitochondrial DNA mutation using more than 200 non-small cell lung cancer or melanoma clinical sample. We found 40% patient, about 40% of patients have mitochondrial DNA mutation, high allele frequency, more than 20%. It's a little strange. We could not find the hotspot mutation. It's a little strange mutation data. This is summary non-small cell lung cancer cohort. Smoking status are little related to mitochondrial DNA mutation in one cohort. However, another cohort not related. This is melanoma cohort summary. UV-related melanoma. Please look at this column. UV-related melanoma, head, neck, or dorsal surface melanoma tended to have a higher frequency of mitochondrial mutation. Because UV induced such mutation, it's very reasonable data. And the PD-1, we evaluated PD-1 blockade efficacy. Patient with mitochondrial DNA mutation tended to have shorter PFS and OS by ICI. So we concluded mitochondrial DNA mutated dysfunction mitochondrial transfer impaired T cell function, anti-tumor immunity, and memory formation. And PD-1 blockade efficacy, especially durable response, could be impaired such transfer. We also evaluated cytotoxic chemotherapy efficacy, but comparable data. So such mitochondrial transfer affect especially ICI efficacy, anti-tumor immunity. There's several questions, opposite transfer. We also check opposite transfer, about 10% transfer from surrounding immune cell to cancer cell. And there are recent paper showing from cancer cell to fibroblast transfer induce CAF formation, inflammatory myofibroblastic CAF transformation. We also evaluated other cells in the tumor microenvironment, and we found myeloid cell dominantly receive mitochondria. We confirmed myeloid cell tended to receive mitochondria in the tumor microenvironment using flow cytometry. and mutated transfer to myeloid cell induce myeloid cell macrophage transformation. And we identify specific gene elevation. So we are now creating conditional knockout mouse and analyzing phenotype and anti-tumor immunity. This is very preliminary data in mutated tumor model. Conditional knockout inhibit tumor growth and reduce pulmonary metastasis. However, in wild type model, conditional knockout did not reduce tumor growth, and in this model, lung metastasis is very rare, and no change after conditional knockout. Today, I cannot expose this specific molecule-specific gene. We are now analyzing these mouse model, and I'd like to present in the future these data in the future. I appreciate every collaborator, especially Nature paper first author, PhD student, Dr. Ikeda from Chiba University, and final talk myeloid cell data, PhD student Dr. Watanabe and Dr. Omae, and Dr. Kawazu and Dr. Mano, sequence collaborator. And Dr. Inozume gave me a fresh clinical sample. Thank you for your attention.
Okay, thank you so much, Dr. Togashi, for a wonderful talk. So now open for question. Okay, Zobang.
This is really a beautiful talk. I have two questions. The first question is, in terms of this transfer of a tumor cells mitochondria to whatever immune cells, how many, is it general phenomena? Have you tested multiple tumor cells that can do the same?
Thank you for your question. I evaluated various cancer cell type, breast cancer, lung cancer, melanoma, and RCC. Of course, depend on transfer frequency varied, but generally occur.
Okay. I have second question is really most interesting. You see, essentially, the donor mitochondria now displays the recipient, right? The recipient mitochondria. So have you tried just use direct fusion, using like a direct fusion rather than using some kind of undefined transfer mechanism? The question is whether or not the direct transfer is something else happens to enable this donor mitochondria to do something that's escaped from the mitophagy, for example.
You mean cell fusion or
fusion, right? You can fuse your tumor mitochondria, right? Tumor cells with the lymphocytes, and you can ask whether the lymphocyte mitochondria can be displaced. That's what you show.
Unfortunately, we did not evaluate fusion. It's an interesting point, but we did not evaluate.
Okay.
I wonder if the mitochondria DNA transfer is restricted to TIL, or does it occur to stroma cells or non-T cells?
Thank you. In recent Nature Cancer paper, I show cancer cell transfer to fibroblast mitochondria. However, in our mouse model, such transfer relatively rare, and myeloid cell tended to receive mitochondria. So we focus on myeloid cell transfer.
Okay, question. Yeah.
Thank you so much for the comprehensive talk. Thank you. I was just wondering that many metabolic genes has like a redundancy, two different systems in the cytosol or the mitochondria. Have you ever tried to see if after the TNT is there a different metabolic gene signature or a different metabolite from that? Because we all see there's many like RS or the glycolysis pathway enriched. Thank you.
Thank you. In previous Nature paper, we did not evaluate such comprehensive metabolism or transcriptome. However, now analyzing CD45 positive immune cell comprehensively, and we identified the metabolism change after transfer and gene expression change after transfer, and we also identified the epigenetic change after transfer. It's very interesting data.
Okay, Dr. Go.
Yeah, thank you for your talk. Do you think the exosomal transfer is a major mechanism or the nanotube transfer?
In T-cell data, in previous our T-cell data, exosome and TNT both important. However, many published data show TNT could be more important. In our present mild cell and cancer cell interaction data also show similar tendency. TNT very important. However, maybe depend on cell type, I think.
More question? We still have almost 1 minute. Question. So if there's no question, I can have a question for you. Yeah, Dr. Togashi. So wonderful talk. So basically, you show this mutation, the tumor mutation in mitochondria seem to induce ROS, right? And this ROS seem to actually promote this type of mitochondria in T cell. Actually, go through this hormone type of replacement. So my question is, for those tumor without a mitochondria mutation, do you think that let's also have a similar type of the transfer event happening, or you think this mutation acts through the ROS to drive this process?
Thank you. I also trace wild type mitochondria using SNP. No functional SNP, and we also confirm SNP also transfer to immune surrounding cells. So maybe mitochondrial transfer frequently occur in the tumor microenvironment, but wild type not so such have effect, but mutation have affect such T cell or mild cell function.
Okay, so the last one is that assuming that this mitochondrial DNA mutation will eventually cause a mitochondrial dysfunction, right? So meaning that if you have a specific mutation event occur in mitochondria, so that it probably is critical for leading to a mitochondrial dysfunction. And I think tumor cell like to have a defect in mitochondria. Yet in a T cell, you need the beta oxidation to drive the T cell activation. So that's and I know in the future you are doing a specific DNA mutation. So in other words, it's a random event of mitochondrial DNA mutation can also drive the same thing, or you have to have a certain mutation eventually cause of the mitochondria, dysfunction in order to have all these transfer event occur. So you know my question, right?
I evaluated some of the mutation, our identified mutations, but not all mutation. And I have previously shown a little no hotspot in mitochondria DNA mutation. So maybe some mutation dysfunction and other mutation no function. But we we cannot discriminate function and dysfunction now. It's in the future we should address such issue.
Okay, thank you. Due to the time issue, we'd like to conclude this section. Please welcome me to give Dr. Togashi another round of applause. Thank you.
Thank you so much.